Hey class, the next video in our series of notes on cell division and DNA unit is going to be our part 4, and we're really going to look at what RNA is and its role in the process of making proteins and specifically transcription in this set. So to begin with, we need to talk about what RNA structure is before we talk about transcription, so we understand RNA's importance as a molecule. RNA stands for ribonucleic acid, so it's the same. It's similar sounding as DNA, obviously, but instead of deoxyribo, it's just ribo nucleic acid, and that has to deal with the structure of the sugar, which we won't necessarily get into much this year. The idea that the sugar is a little bit different. All right, it is single-stranded. That's a really important concept to understand, that when we refer to DNA, we're always talking about two strands in that anti-parallel nature. When we start talking about RNA molecules, we're talking about just one single strand. There's no complementary pair to it. RNA is a special copy of DNA, basically used to make proteins. So we go from DNA to RNA, and we'll get into that process in more detail. What you need to know is that RNA is made of nucleotide, just like DNA, only it varies in a couple of ways. So it's made of this nucleotide, all right, just like DNA. Has a phosphate group, same exact thing. Phosphate group is right there. The next difference, it's made of a sugar. That sugar is drawn in that pentagon shape too, but instead of it being a deoxyribose sugar, it's just a ribose sugar. That just changes the oxygen in the molecule, really. That's what we're referring to. But otherwise, everything else really looks the same. The next biggest difference is the nitrogen-containing base. DNA had A's, T's, C's, and G's. RNA has A's and G's and C's, just like DNA, exactly the same. The biggest difference is that the thymine molecules don't exist in our RNA, and then that we have this base called uracil, which replaces a T, and we'll see where that comes into play. Complementary base pairs covered up by our picture here, but the idea that anytime that we have A's in a molecule, a uracil is going to be replaced. So if a DNA strand is red and RNA is being made from it, every time they come across an A, instead of sticking a T with it, they would place a U. Otherwise, all the base pairing rules still apply for making the RNA. There also are 3 types of RNA that we're going to talk about through the process of finishing this unit up. There's mRNA, which is referred to as messenger RNA. It's a messenger. It carries a copy of the DNA's instructions. mRNA is what's going to be created by the end of this process. All right. rRNA is ribosomal RNA. We'll really refer to rRNA and tRNA in the next set of notes here when we're talking about the next process that occurs. Basically, rRNA makes up ribosome, so it's ribosomal RNA. This is a big structure where proteins are assembled. So we talked about the cell, and we said ribosomes help make proteins. Well, this is kind of how they do that. tRNA is transfer RNA. This carries amino acids, which also help build proteins. The amino acids are just the small building blocks of proteins, and that'll come in the next set of notes as well. So to begin with, transcription process here. Understand that transcription is this process that we see up top. We have DNA being turned into an RNA molecule. So that's what transcription is doing. We're basically taking a complex process and breaking it down into 2 steps for notes here, just so you can see what's going on here. Step 1, RNA polymerase is an enzyme. It lays down RNA nucleotides according to the DNA sequence to create a single strand of RNA. There's that single strand again. And then the step 2, the new bases are added according to the base pairing rules, just like we talked about in the previous slide. Anytime, if we have a DNA molecule, our RNA polymerase will jump on top of this molecule right here. So RNA polymerase will jump on there and start running down the molecule with it and spitting out an RNA strand every time it comes across a certain letter in the DNA strand. So it starts running across. Anytime it sees an A, it's going to spit out an RNA-U. Anytime it sees a T, it's going to spit out RNase A. Anytime it sees a T, another A. Anytime it sees a C, another G. So you see that process unfold there in this animation too. So if we look over here, that's an RNA polymerase. The thing spitting out the red line behind it is the RNA molecule, single strand. So it flies along this double-stranded DNA molecule. It reads one side of it, and every time it comes across a base in the DNA strand, it creates a single-stranded RNA molecule coming out the back end there. So there's our RNA right there, out the back. So RNA polymerase just reads it, reads the DNA molecule, and creates an RNA strand from it, which is instructions for making proteins.